Welcome to EPG Pathshala on the subject Social Medicine and Community Health. I am Professor C.P. Mishra, working in the Department of Community Medicine, Institute of Medical Sciences, Banaras Hindu University. I shall be discussing the module Endemic Fluorosis. This module belongs to the paper Environmental Health. This module has been developed by a team comprising of myself as principal investigator. The paper has been coordinated by Professor Dr. B.M. Gupta, ex-professor and head, Department of Community Medicine, IMS BHU Varanasi. The module has been written jointly by myself and Dr. Jaya Krishna of the from my department, and the module has been reviewed by Dr. J. Tandon, ex-professor and head, Department of Community Medicine, Institute of Medical Sciences, Banaras Hindu University, Varanasi. The learning objectives that has been set for this module are, one should be able to state properties of fluorine and its role in human health, mention recommended level of fluoride in drinking water, define fluorosis and enumerate its types, state magnitude of fluorosis worldwide and in India, enumerate agent, host and environmental factors of fluorosis, enumerate features of skeletal and non-skeletal fluorosis, and state criteria of endemicity of fluorosis. After this, one should be able to list methods of prevention of fluorosis and describe salient features of National Program for Prevention and Control of Fluorosis. As far as properties of fluorine is concerned, it is the 13th most abundant element in the Earth's crust. Most electronegative of all the elements. Emitted from volcanoes as gaseous fluorine, which reacts immediately with water to form hydrofluoric acid. Mainly found in rock and soil in various forms of barely soluble calcium fluorides. Concentration in sea water is about 1.2 mg per liter, that is the parts per million. Concentration in fresh water ranges from near 0 to 25 ppm in the Rift Valley of Tanzania, and worldwide freshwater fluoride concentrations are generally less than 0.1 ppm, but commonly up to 2 ppm in some areas. Recommended level of fluoride in drinking water. Recommended level of fluoride in drinking water is around 0.5 to 0.1 milligram per liter, and up to 1 ppm is drinking water is desirable. If fluoride level in drinking water is below 0.5 milligram per liter, it leads to dental caries, and above 2 ppm level in drinking water leads to dental fluorosis and skeletal fluorosis. The World Health Organization has set the standard of fluoride in drinking water at 1.5 milligram per liter to be adopted by nations. Fluorine plays an important role in human health. It is concerned with the growth of the skeletal system, and it is one of the important components of teeth and bone. Therefore, if there is a deficiency of fluoride, it will lead to dental caries, that is decay of teeth, and if there is excess, it leads to a condition known as fluorosis. So it is a dangerous in both ways. It is called double-edged sword. It means that less is also dangerous and excess is also harmful. Dental caries is a condition that is often related to diet-related. It is a diet-related disease in terms of both total cost and healthcare cost, if we consider, it is important diet-related disease. There are evidences to show the potential for health promotion at the population level to target dietary sugar restriction as common risk factor approach. Approaches for prevention of dental caries: fluoride toothpaste. The overall caries inhibiting effects derived from pooled result of 70 trials, 24% protection, and the effect was independent of background domestic drinking water fluoride status. Fluoride varnishes for preventing dental caries in children and adolescents. Pooled prevented fraction estimate is around 33%. The confidence interval ranges from 19% to 48%. Role of diet in caries prevention: for a given dietary challenge, risk of caries at any tooth site will depend upon plaque bulk, access to saliva, and fluoride exposure. So three factors are operating, and from this diagram we can appreciate that if area is shielded from saliva, if there is insufficient use of fluoride, and if there is insufficient oral hygiene, the pooled effect of the three phenomena increases the risk of caries tooth. So, better access to saliva, better oral hygiene, better use of fluorides, that will reduce, and dietary changes that will reduce ultimately the chances of caries. For a given dietary challenge, risk of caries at any tooth site will depend on fluoride exposure, plaque bulk, and access to saliva. So, we just discussed that the deficiency of fluoride in drinking water leads to dental caries, whereas excess of fluoride in drinking water or in food substances, excess consumption of fluoride in any form can lead to fluorosis. So fluorosis is a disease caused by deposition of fluorides in the hard and soft tissues of the body. It is not merely caused by excess intake of fluoride, but there are many other attributes and variables which determine the onset of fluorosis in human population. It is usually characterized by discoloration of teeth and crippling disorders. There are different types of fluorosis depending on the area of involvement. It can be dental fluorosis, it can be skeletal fluorosis, and it can be non-skeletal fluorosis. So there are three types of fluorosis depending upon the principal organ involved. Dental fluorosis: children living in high fluoride zone are bound to get dental discoloration, which may be seen even in the deciduous teeth. So change in dental coloration, discoloration is the first manifestation. Initially, glistening white teeth becomes dull and yellow-white spots appear on the surface of teeth. Gradually, these spots turn brown and presents itself in a brown streaks which are closer to the tip of the teeth. In late stages, the whole teeth becomes black. Teeth may be pitted or perforated and may even get chipped off. In endemic zones, people lose their teeth at an early age and may become edentulate. Skeletal fluorosis, it affects young as well as the old. The symptoms include severe pain and stiffness in the backbone, joints, and a rigidity in the hip bones. X-ray examinations of the bones reveals thickening and high density of bones. In some patients with calcium deficiency, osteomalacia type changes are also seen. Constriction of vertebral canal and intervertebral foramen, there results in pressure on nerves leading to paralysis. As far as non-skeletal processes are, there are several manifestations. There are convincing evidence of involvement of skeletal muscles, erythrocytes, gastrointestinal mucosa, ligaments, and spermatozoa on consuming more than optimal intake of fluids. Detection of fluorosis at early stage is possible by understanding the soft tissue manifestations. In the fluorosed muscles, actin and myosin filaments are destroyed, and mitochondria lose their structural integrity, thereby providing evidence of depletion of muscle energy. The erythrocyte membrane loses its calcium content in presence of high fluoride. Non-ulcer dyspeptic complaints are manifested by consuming high fluoride in water and food. Infertility due to oligospermia and azoospermia is commonly seen in fluorotic belts. Hydrophorosis is a condition that refers to waterborne fluorosis. Due to excess of fluoride in water, it results in hydrophorosis. Foodborne fluorosis, if food chains contain high fluoride, it results into foodborne fluorosis. There is a term called industrial fluorosis. Industrial workers engaged in industries which use fluoride in their manufacturing process, and population nearby are the victims of fluoride toxicity. This is known as industrial fluorosis. There are a number of industries which are using fluoride. For example, aluminum industry, automobiles, vehicles, bricks industry, chemical industries, enamel industry, fertilizers, glass and ceramics, oil refineries, plastic industry, pharmaceuticals, pottery, refrigeration industry, rust removal industry, and steel industry, toothpaste industry, and welding. So a number of industries are there which are using this. There is a term called neighborhood fluorosis. Industries using fluoride may discharge effluents into streams of rivers and pollute the groundwater or surface water. People living nearby these industries who depend on groundwater may suffer from fluorosis. Now, coming to the magnitude of fluorosis worldwide, fluorosis is worldwide in distribution. It is endemic in 22 countries. That means that the disease is habitually present in these areas. By endemicity, we mean that Asia, and in Asia, India and China are worst affected. Mexico and Northern Argentina in Latin America, they are the principal affected countries. East and North Africa are also endemic for fluorosis. As far as occurrence of fluorosis in India is concerned, Rajasthan state and Gujarat state in North India and Andhra in South India are worst affected. Punjab, Haryana, Madhya Pradesh and Maharashtra are moderately affected. Tamil Nadu, West Bengal, UP, Uttar Pradesh, Bihar and Assam are mildly affected. Throughout India, fluorosis is essentially a hydrofluorosis. It means that is caused by excess of fluoride in drinking water, except in parts of Gujarat and Uttar Pradesh, where industrial fluorosis is also seen. In worst affected states, maps have been prepared of geographic pathology on the basis of fluoride distribution in the drinking water. Now, fluorosis has a lot of socioeconomic implications. In India, social and economic implications due to fluorosis are enormous because of widespread crippling effects, disability, and decreased work capacity. So, if a person is crippled, disabled, and has reduced his working capacity, certainly his employment potential is decreased and his earning capacity will also decrease. That will result ultimately less economic generation for him, and which ultimately will result into poverty. So this is one of the important socioeconomic implications of fluorosis in India. Discoloration of teeth leads to social problems, particularly in children. As far as epidemiologic triad is concerned, we can discuss as agent factors, host factors, environmental factors. The agent factors are primarily it is a fluoride which is present in drinking water in excess amount, more than permissible level, and that results in fluorosis. When fluoride in water is more than 1.5 milligram per liter, it is toxic to health. pH in terms of alkalinity of water promotes the absorption of fluoride. Calcium in the diet reduces the absorption of fluoride. Hard water rich in calcium reduces the fluoride toxicity. Fresh fruits and vitamin C reduces the effect of fluoride. So it has a protective effect. Trace elements like molybdenum enhances the effect of fluoride. Now, a number of host factors are there. In school-going children, we often see dental fluorosis. In third and fourth decade of life, we mainly see skeletal process, or they are victims of skeletal process. Males suffer more from than females. Illiterate suffer more frequently in the fluoritic belts. When aluminum ores are mined, it is seen as an occupational health hazard. And there are a number of industries where this may be a problem. As I have discussed earlier, a number of environmental factors are involved. These are high annual mean temperature, low rainfall, low humidity, fluoride-rich natural subsoil rocks, vegetables from high fluoride belts, fluoridated toothpaste, particularly when used by children, tropical climate, and particularly in developing countries, the environment is so conducive that it leads to fluorosis. There are a number of risk factors of fluorosis. Lack of awareness about the role of fluorine in human health. People need to be educated that when there is a fluoride deficiency, it may lead to dental caries. When there is a fluoride excess, it may cause skeletal fluorosis, non-skeletal fluorosis, and dental fluorosis as well. So excess and less intake both could be harmful. Increase of population and industrialization leading to shortages of safe drinking water. The water which is safe for human consumption is gradually declining because of rising population, because of many industrialization processes, and excess of fluoride in water does not change the color of water or alters its taste. This is a big drawback. If the color, if this could have changed the color or it could have altered the taste, probably people have avoided its consumption. But since this is not being caused, thus the consumer will not know unless frank stage of fluorosis is produced either in teeth or in bones. So generally we'll find that the most of the cases of fluorosis are detected late when advanced signs appear, and therefore it poses a very serious public health problem. Groundwater supply having excess of fluoride content is a very significant risk factor. Food habits of consumption of excess black tea, tobacco, and black salt also results in this. Pregnant lactating mothers and children are most vulnerable, and undernutrition can potentiate the effect of fluoride in causation of fluorosis. Now, after discussing certain epidemiological features in terms of who's affected, where disease is more common, when people get affected, and what is happening, with the passage of time, the fluorosis problems are increasing globally as well as in India because of shrinking water sources, which is a protective for human health, or safe water supply. As far as prevention of fluorosis is concerned, as in all diseases we discuss generally in the three headings: primary prevention, secondary prevention, and tertiary prevention. Primary prevention are those measures which are instituted so that the fluorosis problem will not arise in a particular area. This includes one of the most important things is the health education. People should be informed about adverse consequences of fluorine. Second is capacity building for health promotion and prevention of fluorosis, particularly of the capacity building of the health and related staff, so that they can educate the people about prevention of fluorosis and other health promotional measures. Defluoridation of water in those areas or those water which has high fluorine content. It is necessary that defluoridation efforts are initiated in that area. Defluoridation of water, there are precipitation methods, there are adsorption, ion exchange methods, and domestic defluoridation. Among the precipitation methods, the most popular methods are lime treatment, alum treatment, and Nalgonda technique. Nalgonda technique has been developed by Indian scientists. It is based on the amount of fluorine in the drinking water and alkalinity of the water, expressed as magnesium calcium carbonate. The amount of alum to be mixed with water is calculated based on considering these two factors. Activated alumina, defluoridation of drinking water by activated alumina is the method of choice in developed countries. Domestic filters using activated alumina have been accepted by the users. Use of proper grade of alumina, activated alumina, is a must to obtain satisfactory result. Regeneration of exhausted activated alumina should be done after 4 to 5 months. Domestic defluoridation technique is also there. Say for example, if you want to purify 40 liters of raw water, for defluoridation of water at domestic level, first of all the local women needs to be trained about the process of defluoridation. And suppose 40 liters of raw water containing 3 ppm, parts per million fluoride content, and alkalinity of 200 parts per million. We'll add to this 6 ml of lime solution, which contains 10 gram of quicklime in 1 liter of water, plus 120 mg bleaching powder, plus 120 mg of alum solution. 100 gram of alum dissolved in 1 liter of water. This will bring down the fluoride content to a level of 1 ppm, which is acceptable by all standards. So domestic defluoridation of technique can be employed. What is needed that the women's capacity in this regard must be enhanced by proper orientation and training. Early diagnosis and treatment is the second stage, so that if fluorosis occurs in a particular area, it should be detected at the early stage. And that requires, number one, capacity building of health personnel, personnel in early diagnosis and prompt treatment. Identifying skeletal fluorosis on the basis of signs and symptoms and using following tests: Quant test, chin test, stress test. Now, as far as clinical features of the skeletal fluorosis is concerned, we have discussed earlier, but just to reinforce once again, there will be vague pains and aches in extremities and joints. In fluorosis, the joints become stiff and flexion of joint is difficult. X-ray changes, new bone formation and calcification of and tendons and ligament takes place. Tests for skeletal fluorosis. Affection of the joints can be ascertained through simple tests which can be carried out at the bedside and in the field. One is Quant test. The subject is asked to lift a coin from the floor without bending the knee. So, a fluorotic subject would not be able to lift the coin without flexing the large joints of the lower extremity. So, this is a first and foremost test which can be done in the field situation, and that clearly demonstrates that there is a problem in the flexing of the lower joint. The second test, which is very important, is chin test. What is done here? The subject is asked to touch the anterior wall of the chest wall with the chin. If there is pain or stiffness in the neck, it indicates presence of fluorosis. And third is stretch test. The individual is made to stretch the arm sideways, fold at the elbow and touch the back of the head. The point of importance here is that one should not bend the arms. When there is pain and stiffness, it will be not possible to reach the occiput, indicating presence of fluorosis. So workers can be trained at the field level, and they will be able to detect skeletal fluorosis by these simple tests in the general population: Quant test, chin test, and stretch test. The point of importance is that in any area, in a given area, endemicity of fluorosis can be identified by the criteria when more than 1/5 or 20% of the persons surveyed in a high fluoride area shows positivity of clinical tests just enumerated, means Quant test, chin test, or stress test. And by applying these tests, if 20% of the population shows positivity of the clinical test just enumerated, indicates endemicity. A number of diagnostic tools can be applied to detect the fluoride level in drinking water, in blood serum, urine spot urine samples, and radiograph of the forearm. And in order to treat these cases, nutritional interventions can be planned. These nutritional interventions could be administration of vitamin C and other antioxidants, which may help in reduction of the progression of the disease. Tertiary prevention is the last stage when the disease cannot be detected early. This may lead to further problems in terms of disability, and it may require rehabilitation initiatives. So, capacity building of secondary and tertiary care hospitals at district level and at medical college level for reconstructive surgery and rehabilitation can be attempted. Coming to the last point which we would like to discuss, that Government of India has been very serious about the problem of fluorosis. There are certain pockets, certain areas in the country where fluorosis is of very serious magnitude. And in those areas, most of the water sources have been tested for fluoride content, and they have been labeled not for fit for human consumption, but the red mark has been given on them. In fact, National Program for Prevention and Control of Fluorosis in India has been taken as a sub-mission of Rajiv Gandhi National Drinking Water Mission, which is a very large mission for water supply. And in Rajiv Gandhi National Drinking Water Mission, there was an attempt to provide safe water in adequate amount in terms of quantity. But the other important dimension has been also taken into consideration very recently, that the water that is being supplied should not be harmful to the human being. And one of the major criteria is that the fluoride control or fluoride level in the drinking water should not be high. Surveillance of fluorosis in the community was considered a major activity. And surveillance, it means if cases are occurring in the community of either dental fluorosis or skeletal fluorosis, that is a clear-cut trigger that the soil and subsoil and water that is being supplied to the community is not safe for human consumption. So surveillance activity is a major activity, not only of surveillance of fluorosis, even the water sources and those sources which are found not very conducive for human consumption should be stopped. It has been a 100% centrally sponsored program. The program which was launched had many objectives. Some of the most important objectives are to assess and use the baseline survey data on fluorosis supplied by Ministry of Drinking Water and Sanitation, and comprehensive management of fluorosis in selected areas, and capacity building for prevention, diagnosis and management of fluorosis cases at different levels of care, be it primary, be it secondary, be it tertiary, depending on the needs of the population. Guidelines of programs have been set to identify and label any potable source safe and educate the population to conserve and use the source for drinking water and cooking only. To tap safe, low fluoride aquifers, to arrange for blending of water from different sources to obtain adequate quantities of safe water, and to supply this potable mixture through designated safe stand posts. This is very important because some of the water sources may be having a high fluoride level, some of the water sources may be having less fluoride level, and since fluoride is an element that whose deficiency and excess is both harmful, by this blending procedure, the fluoride level in drinking water can be brought to within the normal safe range. To provide piped water supply schemes through distant sources, because distant sources are less likely to be contaminated by human activities or industrial activities. Rainwater harvesting techniques have to be used to mitigate fluorosis and to provide community treatment plants to remove excess of fluoride. So community treatment plant, particularly in those areas where fluorosis is a very significant problem, and in those areas it is desired that the defluoridation of water should take place at community level and also at individual level. Finally, coming to the summary part, would like to conclude the entire discussion. We identified that fluorosis is an important problem in many parts of the world. And there are many areas in the country, particularly in our country, India, where fluorosis has become endemic. Not only endemic, but it is becoming a serious problem nowadays. Onset of fluorosis is not determined by fluoride, but a large number of agent, host and environmental factors determine its occurrence. So certainly we have to take care of those for fluoride content, or we have to identify those water sources which have high fluoride level, and initiatives has to be taken to bring the fluoride content in them below a particular level. Characteristic features are for the disease is discoloration of teeth, and ultimately other changes in case of dental fluorosis, and skeletal fluorosis results in crippling disorders. But there are simple tests which can be applied at the initial stage in terms of Coin test, in terms of chin test, in terms of stress test, which can be applied by paramedical staff at the field level, and endemicity of the disease of fluorosis can be established if out of 5, one is a victim of fluorosis. Although fluorosis once established is irreversible, but it can be prevented through simple interventions. Throughout India, fluorosis is essentially hydrofluorosis, except in part of Gujarat and Uttar Pradesh, where skeletal fluorosis is also seen. So there are a number of measures at primary prevention, secondary prevention, tertiary prevention, through which this disease can be taken care of. Thank you very much for visiting EPG Pathshala.